the presentation for us.
Yes, working. I'm not quite sure the position.
Oh, yes, something.
No, the other way. Over your ear.
Okay.
Okay. You here? Clear? Yeah. Okay, thanks. It's a pleasure to be here. I'm back again after 2 years. I was contributing to this course. I'm going to talk about dopamine signaling. I work on signal transduction in the central nervous system, and particularly phosphorylation and dephosphorylation reactions. And when I was preparing this talk, I realized I was going to be talking on the first day. And usually I talk at the end of the course, so everybody knows about dopamine by then. I was like, I'm going to have to explain why drugs and dopamine go together. But fortunately, the students and postdoc Anna and Emma have already introduced you to the general area that I'm going to talk about. So I thank you for your introduction. You could probably just give this lecture. Basically, before I start, I just want to thank people who are connected also to the Belgian lab, Jean-Antoine Giraud, who I've worked with for many years, and I'm going to talk about some data shared with him and also from the Belgian studies. And I also stole some slides from them, as well as I stole some slides from Jim Sermeyer, with whom I also worked for many years. So thank that. And the work that we do is supported by NIDA. You'll hear from Nora Volkai, I think, later in the week. And we are also supported by NIH through NIMH and the Department of Defense in the US. And they support, NIDA in particular supports the Neuroproteomics Center that I direct at Yale University. Okay. So I'm going to try to do a few things. I'm going to basically stick to pretty much all published work. So if there's 2 or 3 papers, and if you need the references, I'm happy to give you the names or the senior authors are on the slides, but it's pretty much published work. And really, I just want to go through the details of dopamine receptor signaling, particularly talk a bit about how G-coupled receptors work, how they generate second messengers like cyclic AMP. Then spend a fair amount of time, is really the core of the talk is on how proteins are phosphorylated and dephosphorylated, and how some of the molecular details of how that works. And then you already heard a little bit about this protein earlier from the student's talk. This protein DAR32, which we think is a sort of a key integrator of signaling in striatal neurons, and as such is a key target for dopamine. And I'll tell you a little bit about how we think that protein works and how it functions within the actions. Specifically, I'll show examples from several drugs, but mostly the work is from looking at the effects of psychostimulant cocaine. And then, again, this topic has already been briefly brought up, but what are the challenges in terms of studying signaling in specific cell types in the CNS, which seems to be an important area for many aspects of study of the nervous system at the moment, but particularly when it starts to work at the protein level and how protein signaling is changed, because the signals appear to be very specific and discrete for one particular neuron, and next door neuron is changing, either not changing or sometimes changing in a completely opposite direction. And so how do you interrogate those sort of changes? Okay, so I'm a biochemist by training, and so when I think about these various topics in the CNS, I try to be as quantitative as possible. So some of the questions that I'd like to sort of address, and at least hopefully you remember some of the details, is things like in terms of deep dopamine receptor signaling, how many GPCRs there are, and how many different types of dopamine receptors, how do they work, how many different types of G proteins there are, secondary messengers, what are they, how many, in terms of protein phosphorylation and dephosphorylation, how many kinases there are in the human genome, how many phosphatases, and how these numerically relate to each other. And then we read the topic of protein phosphorylation has already been raised, but you know, how many proteins are phosphorylated in a typical neuron in response to a drug like cocaine? What's the stoichiometry of the phosphorylation? What's the specificity of signaling? And then, you know, some more, I'm going to probably start with just some sort of general comments. You know, how many genes are we talking about that are, whose expression might be affected? That relates particularly to, again, the topic was brought up that new proteins may be synthesized in response to psychostimulants. So how many proteins, mRNAs, etc. And then maybe even drill down to, you know, when we're talking about changes in synaptic function, in terms of dendritic spines, I mean, how many proteins are there, how many receptors, how many channels, how many kinases, how many phosphoproteins. So those are some of the questions I'm not going to address directly, but those are the sort of questions that when I'm doing my own research, that I try to consider, because I think it's important to get to be as quantitative as possible. So just for example, just to see where we all are, I mean, how many GPCRs are in the human genome? Does anybody have an answer to that question? 10, 20, 100?
1,000.
1,000, okay. And why is 1,000? Why is the number 1,000? What's the predominant GPCR that makes up 1,000? Odor receptors, exactly. So we're going to talk about dopamine receptors. How many dopamine receptors are there? Main types? 5, 6, 5. 5 main types. Okay. Let me see how else we have. So how many kinases are in the human genome? Do you have any idea? Thousand, 100. 1,000. That's pretty close. How many genes are in the human genome?
Depends on the day.
And Marina, how many genes do you expect?
32,000 is the last count, I think.
It's a little bit high. It's probably more close to 20
29?
something.
25.
20,000. So yeah, so it's about 500 kinases. So 1,000 is pretty good. How many protein phosphatases do you think there might be? The same. So it turns out there's a lot less phosphatase genes than there are kinase genes, and I'll try to get into that a little bit. Okay, how many proteins are phosphorylated in a typical cell? Out of the 20,000 genes.
20,000?
Sorry?
20,000?
Probably almost every protein in the cell is phosphorylated at some point. And in fact, most proteins are phosphorylated at multiple sites of phosphorylation. As we heard for S6 protein, there are 5 sites. But in fact, that's not atypical. Some proteins get phosphorylated on, you know, the protein, phosphoprotein tau, for example, which is goes wrong in Alzheimer's disease, is probably phosphorylated on about 100 sites. So when you think about what I'm going to tell you, basically anything I say today is just a very, 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 very tip of the iceberg in terms of what we think about cell signaling. So when we talk about exposure of drugs of abuse to a particular cell type, there are many, many things happening that we actually don't know about yet. We can interrogate a few that we have probes for, but the reality is it's part of a huge network of signaling, and trying to understand that, and the complexity and the overlapping consequences of hitting different pathways is a really major challenge, and we're really nowhere close to that even for a single cell type. For example, a cancer cell type, we have some ideas of one sort of phenotype. But when we talk about hundreds and hundreds of different cell types within the CNS, that we really have very little information. So there's a long way to go before we really understand this area. Okay, so basically what we're really talking about here is linking behavior to the environment and genes. And if we look at how this happens, we have a situation where gene expression leads to changes in protein levels. The level of intracellular signaling, which is where I'm going to focus, occurs on the protein level. This changes in intracellular signaling modify synaptic and membrane proteins, affect local circuits, neuronal assemblies, and larger neuronal populations to ultimately influence behavior. And what we're looking at in terms of addictive drugs, no matter which ones we're talking about, what we're really talking about is how they perturb this part of the story here, this intracellular signal. And if we think about the time frame of that, we're really looking at time frames that start at the millisecond stage, move through seconds to hours to days, months, and longer in terms of addiction. And so how are these sort of the initial exposure to a drug of abuse, which may only occur for a very short time, how does it ultimately affect these longer-term processes that occur in the brain, and how they maintain for many decades. In some cases, people become addicted to nicotine or whatever when they're young, and they will retain that addiction through their lifetime. So initially, when we think about the very fastest things that happen, we're talking about very quick changes in protein conformation that would be associated with electrical activity in the brain. As we change and think about how things are modulated, really those are happening within seconds to minutes. And this is where the area of protein phosphorylation is particularly important. As we shift to more stable changes in terms of synaptic plasticity, what we're talking initially are changes in protein trafficking, so protein localization within the cell, receptors being moved from reserve position to the synapse. And then eventually over an hour plus or to many hours, we're talking about stable changes, often in transcription events that lead to stable changes in protein synthesis that cause changes in neuronal growth and changes in morphology that can last for many days, months, and years. And when we start thinking about dopamine receptor activation, which is the target of many drugs of abuse, what we're really talking about are events that happen within probably, beginning within less than a second of engagement in these receptors. And these changes can then have consequences that last for a long time. And so basically what I'm going to talk about are some of the mechanisms that are initially engaged and how they are mediated through this initial phase. So the brain is complicated, we all know that, and dopamine we've already had a little bit about in the 3 presentations this morning from the students, where dopamine, midbrain dopamine is projected. I'm going to focus most of my attention to changes in biochemistry in the dorsal striatum and a little bit in the ventral striatum to make things a little bit more simple. But obviously dopamine projects too and modulates processes in many different parts of the brain which are connected to each other. In the human brain, there are about 500,000 dopamine neurons that project from the midbrain to areas such as the ventral dorsal striatum to the prefrontal cortex. We already have about that. In mice, there are about 20 to 30,000, so there are not many of these neurons, but they have vast influence through their extensive arborization and projections. This circuitry is oversimplified, but basically we've already had a little bit about that. These the medium spiny neurons of the striatum, the dorsal and ventral striatum, that make up about 90 to 95% of the neurons in this part of the brain, and they contain within their the medium spiny neurons 2 large subpopulations of about 50% each. That are either the direct projection neurons that are mostly, as we heard already, it contains D1 type receptors, dopamine receptors, that project directly to the substantia nigra. And then a second class of D2 are the indirect pathway neurons that synapse via the globus pallidus to this subthalamic nucleus and then onto substantia nigra. And they're part of a sort of topological loop that is critical for a variety of aspects of motor learning, motor control, motor learning, habit learning, and reward learning. And I won't get into those details. I'll leave that for others to do so. But within these medium spiny neurons, there are a number of proteins which we think are specifically regulated by dopamine that's released by these blue neurons that project from the nigra bract to both the dorsal and ventral striatum. And so what we're really talking about are the synapses between these dopamine neurons, these GABAergic projection neurons, which are in turn excited by these glutamatergic cortical neurons, which are shown here. So if we look at that in a little bit more detail, This is the same schematic, but we expand and we look at the medium-defining neuron in the nucleus accumbens in this case, where we're talking about projections of dopamine fibers onto these the dendritic spines of these cortical of the medium-defining neurons, which are excited through excitatory synapses from cortical afferents. And this is a camera elucidative picture of a particular medium-defining neuron. You can see the cell body, the nucleus, the axon would be over in the other room. And we see it's decorated by these dendritic spines, which are these small protuberances that are shown in the many dendrites of these cells. If you look at in some more detail, this larger magnification, we see are these the dopamine synapses are on the side of the spines, and they're positioned obviously very nicely to release dopamine that can modulate glutamate acting as a neurotransmitter. So from the perspective of signal transduction, what we're really looking at is the ability of dopamine to modulate the signaling of other processes, particularly glutamatergic inputs to these neurons. So we know that the focus of this presentation is obviously related to drugs of abuse, but we've already heard a little bit from Raphael about the potential effects of psychostimulant type drugs in the relationship to ADHD. Dopamine has many other normal and pathological functions. Schizophrenia, there seems to be imbalances between dopamine and glutamate signaling. In Parkinson's disease, we lose the dopamine neurons. They degenerate, particularly in the inputs to the dorsal striatum. And in Huntington's disease, another motor disorder, the output neurons from the dorsal striatum are degenerating. So obviously the work that, any work that we do on dopamine signaling has real importance for a number of different areas related to these various disorders. And so many of the findings that have been made, and particularly signaling within this brain region, have been relevant to other areas as well as drugs of abuse. Okay, so dopamine. So dopamine receptors, how do they signal? Again, we've heard a little bit about that, but there are these two major subclasses of receptors, the D1 and D5 receptor, make up one subclass of dopamine receptor. And they are GPCRs, which are coupled through, in the striatum at least, through this G-alpha S G-off protein, which is the heterotrimeric G protein, as the alpha subunit of the heterotrimeric G protein. It's made up of the beta and gamma subunits. Once in the GTP form, the G-alpha, once ligand is engaged and the GTP is exchanged for GDP, this becomes active. It is able to activate the adenyl cyclase, which can then generate cyclic AMP. And the cyclic AMP's primary target in the cell is protein kinase A. And I'll elaborate about that in some detail. The D2 subclass, in contrast, is made up of the D2, D3, and D4 receptors. It's coupled through a different heterotrimeric G protein, the alpha subunit of which is the alpha I or alpha O. And this has the ability to suppress the activity of adenyl cyclase. But already, the slide is misleading, because in reality, as we've heard a little bit already, the D1 classes of receptor are in a different type of neuron from the D2 class of receptor. So really, we should be, I'm going to jump ahead, we should really be looking at two different diagrams. And so basically, this mostly is occurring, the one subclass, the direct output neurons in the striatum, and the D2 class is actually working with a different set of neurons. So we'll come back to that in a little bit of detail later. So for a little bit of historical context, so the original work that went into describing the ability of dopamine to modulate adenyl cyclase came from work from Paul Greengard and John Kababian in the early 70s. So I worked with Paul Greengard many years, and so it was interesting going back and looking at some of this the original data. But basically, what the work that they did was to describe a dopamine-sensitive adenyl cyclase in the caudate nucleus, or the striatum of rat brain, and realized when they find this very high activity of dopamine-regulated adenyl cyclase, that this had a similar distribution to at that time. We didn't know what receptors there were there, but that by ligand binding to what was termed the dopamine receptor. And so basically, that was the original description of the work. And so they were able to show in this classical paper from Kinase in 1972 that dopamine is able to activate the production of cyclic AMP, and it was highly enriched in these striatal neurons. Okay, we'll just go past that. So basically, the original work was description of generation of cyclic AMP, and subsequently they went on to identify that the target for cyclic AMP, protein kinase A, was highly enriched in the same part of the brain. So the highest levels of protein kinase A are found in the striatum. But in addition to that, the D2 subclass has other features. So in addition of the alpha subunit of the alpha IO tetratrimeric G protein to inhibit the adenyl cyclase, the beta gamma subunits can also function independently to modulate the activity of various ion channels. So there's additional signaling, and I'm not going to talk about it in great detail, that dopamine can engage in it when it activates D2 receptors. And so examples are shown here. So basically, when the D2 receptor is engaged, it can release the active GI alpha subunit that can work on adenyl cyclase. But it can also release the G beta gamma subunit that can directly interact with potassium channels, calcium channels, and sodium channels to modulate directly excitability. So this is important to think about that these are effects that go beyond second messengers and are probably much faster and are specific to the D2 receptor subclass. Okay, so we think I've sort of described specific effects of D1 and D2 receptors, but in fact, it turns out that the situation is probably more complicated than that. And there are a couple of areas. Mark von Zastra, who's one of the organizers of this course, I don't think he's going to be here this week, but has been very interested recently in the sources of dopamine signaling within cells. So we tend to think that the G protein coupled receptors function just at the cell membrane and the plasma membrane in response to ligands from the outside of the cell. But in fact, work that came out, this is from a Neuron paper from a year ago, by playing around with processes that regulate endocytosis, the von Zastra lab showed, and I won't go through the details here, that if they increased endocytosis, they'd actually get a much bigger levels of cyclic AMP signaling once the receptors were actually internalized and moved to sites within the cell rather than being in the plasma membrane. So when we think about signaling through dopamine receptors, it's not just that initial hit, there may be other subcellular consequences of receptor endocytosis that still maintain cyclic AMP generation, and how exactly that occurs is really unclear. Another area of active interest, and you probably can't see the details here, is but is reviewed in this recent article from Susan George, are that in fact these various G protein coupled receptors, like D1 receptors and D2 receptors, but many other types of G protein coupled receptors, when they're co-expressed within the same cell, can actually heterodimerize and function independent in a different way than they would do if they were working on their own. So this is really a kind of another complication of when we think pure receptor mediated events, there may be complexities because they're actually coupling between even D2 and D1 receptors when they're co-expressed in the same cell. And there are numbers of cells in the striatum medius binding neurons that do co-express D1 and D2. The consequences of the signals that are generated are distinctly different than if you just engage one or the other, which is very difficult to biochemically analyze at a population level what the consequences of this is, but it's obviously an area that we have to try to understand. I'm not going to get into that in detail today. But there are also interactions between, for example, adenosine receptors and dopamine receptors, between NMDA receptors and dopamine receptors, and so and dopamine receptors and serotonin receptors. So a lot of complexity going on that we don't have really a good Thomas.
Angus, so how much of that signal do you actually think happens close to the synapses? You had at the beginning a diagram that showed like the dopaminergic inputs right next to the spines, where they receive excitatory inputs.
Right.
Do we have like an idea of what fraction of excitatory inputs gets modulated by dopaminergic inputs, like where all of that occurs?
So I'm going to talk a little bit about that specifically with not quite the context you presented it in, but maybe you can bring it up with. Well, I'll talk about that in a few more slides. Okay. I think it's a timing issue largely. At the biochemical level, what we measure. So it depends how you do the experiment, how you actually measure, right? So if you look at a Western blot, you're homogenizing all the tissue, so you lose any idea of the resolution. If you do it by immunocytochemistry, for example, you may be able to get a hint of where the signal is occurring. But it's probably largely a timing issue because most of the signal that you see generally in terms of dopamine signaling within a few minutes is mostly cell body signal. So somehow the cell has already integrated the signals from these distal dendrites and sent some signal down the dendrite to change the signaling. And that's actually an area which is also an area of challenge in terms of how that signal is maintained and perpetuated. But there has to be initial signaling at dopamine receptors at the synapses, which is then mediated biochemically to the cell body and then into the nucleus, and I'll talk about that in a few minutes. Any other questions? Okay. So I'm going to go through very quickly just to give you a sense that we actually know from a molecular perspective, we know a lot about how these molecules work. So initially this is an area of great expansion at the moment from X-ray crystallography particularly, but we've known about the structure of G-protein coupled receptors from the early work in rhodopsin. We know about the structures of the heterotrimeric G proteins from a number of studies. Here's the G-alpha S and the GDP platform, the beta and the gamma subunits that mediate signaling, as I mentioned, on ion channels. We know that these are attached by lipid attachment to the membrane. We know a lot now about every time you open up Nature or Science, there's another GPCR structure. We know now it's a huge expansion of work. This is an example on the D3 receptor in the presence of a selective D2-3 antagonist. So these structures are really helping to give us more information about how these ligands engage the receptors and how we can design better antagonists and agonists. And so we know a lot about the structural work. This is another example from the adenosine A2A receptor that featured prominently in the indirect pathway neurons. We know the structure of the adenyl cyclase. This is a cartoon. I'll show you the structure in a second. It's an interesting molecule. It has 12 transmembrane domains with 2 catalytic domains. We know that if we look at the individual catalytic domain, it's engaged by the G-alpha S and the GTP platform because of a structural rearrangement that changes the structure of the so-called switch 2 domain that then inserts itself into the target and is able to modulate the activity of the adenyl cyclase to produce cyclic AMP. The drug Forskolin that was talked about earlier this morning, it structurally binds to the active site on the active site of the adenyl cyclase and stimulates directly the production of cyclic AMP. We know about how other aspects of some of these receptors work. This is recent work from Rod McKinnon's lab at Rockefeller. This is looking into the structure of how the beta gamma subunits engage the GERK potassium channel. And so we'll go through the details of this, but you can see from this cartoon the idea that I just mentioned earlier, that the beta gamma are able to be released and within the plasma membrane move and engage this channel and directly negate its activity. And so we have high resolution structures of many of these things, and this is a very rapidly growing area of research. Okay, so that's pretty much it for G-protein coupled receptor signaling. Now I'm going to switch and talk about protein phosphorylation, give you a little bit of background, general background, before moving on to talking about specifically the regulation by psychostimulants. So I already asked this question. There are about 500 and so protein kinases in the human genome. About 428 of them are so-called serine-threonine kinases. So those select serine and threonine within their substrates. There's about another 120 or so which selectively target tyrosine residues, and I'll mention a little bit about tyrosine phosphorylation in a few minutes. But interestingly, in contrast to these 400 plus protein kinases, there are actually only about 20 genes, products that encode the phosphatases that reverse the dephosphorylation of serine 3, which seems on first analysis a little surprising. You'd probably think that it would be more or less equal. But, and remember, as we talked about right in the beginning, there are probably 15 to 20,000 gene products in any given cell type, and almost all of them are phosphorylated. So we're talking about many tens of thousands of phosphorylation sites within cells. And so one of the big challenges in this field is to understand how the kinases identify their selective targets and phosphorylate them, and also how these phosphatases, and relatively few of them, selectively also identify their targets and dephosphorylate, so you can get a coordinated regulation of phosphorylation and dephosphorylation. And so I'm going to talk a little bit about how that works very superficially to give you some sense of how it works. So I already said this, so maybe the 60s, this number says 60 to 70% of proteins are phosphorylated, but it may be more than that. So, so if you're interested in a particular phosphorylation, if you're a student or a postdoc and your PI said, go study, this particular protein, I think it might be phosphorylated. What would be the first thing these days you would do if you were going to study phosphorylation in the brain? So DC, what was somebody worked on DCC? What would you, they said, well, maybe DCC is phosphorylated. What would you? By right, 500 kinases, so you'd have to screen 500 kinases, so it's a lot of work. So actually these days, the first thing you do is go to the computer. Because in fact there are extensive, huge databases out there now that have mapped many, many phosphorylation sites. We're talking about hundreds and hundreds of thousands of phosphorylation sites and hundreds and tens of thousands of proteins. You may have seen in Nature a couple of weeks ago the human genome proteomes was essentially identified. So we know where in many cell types which proteins are being expressed. And so if you have a particular protein, you would go to, there are 3 or 4 or 5 databases that are really, really excellent databases these days. So the first thing you'd go to see, does this protein get phosphorylated? And if it does, which site? So that would be a huge advance. And then, and I'll get into this in a few seconds, but because of the sequences of the phosphorylation sites with the amino acid sequences, you can sometimes get some information about what kinase would be the kinase, and that would give you a start. There are many companies that make particularly cell signaling, and it's related companies that make phosphorylation-specific antibodies. So sometimes these are already available, so you can go study your protein of interest. But so in fact, the field is advanced in terms of what we know is phosphorylated. We know many, many sites. The challenge is still to identify the kinases and the phosphatases and the mechanisms of regulation. Okay, this is just a cartoon of the human kinome, and I'm going to talk about, there are 500 or so kinases here. I'm going to talk about a couple of kinases in the middle part of the presentation, or the end part of the presentation. One is the phosphoprotein, the kinase called ERK, or sometimes called MAP kinase, and we've already heard about protein kinase A and how it's phosphorylase STAR32, but protein kinase A is also important because it regulates other things like the amplification receptor in serine 845 is particularly important in modulating synaptic plasticity, and for example, the transcription factor phospho-Krebs is very important for modulating changes in gene expression. So I mentioned a few other kinases passing, but the pKa and ERK are the ones which I'm going to selectively focus on. So how does pKa work? So pKa consists of a heterotetramer. There are 2, this is sort of, for people who are old enough, sort of 2 Pac-Man type catalytic subunits here. And they're engaged in the basal state and to suppress the activity of the C subunits. These are the R subunits, or the regulatory subunits that are covalently bound to one another by disulfide linkages. And then in the inactive, they engage the active side of the catalytic subunit. And this is a structure shown here, but when cyclic AMP binds the two cyclic AMPs per R subunit, this causes a structural rearrangement in what's called the autoinhibitory domain of the R subunit that removes it from the active side of the C subunit and allows the C subunit to go find the substrate. So when cyclic AMP goes up in cells, this structural rearrangement has to occur. And it frees up the C subunit, which it can then start to move to other places in the cell, which is maybe the first part of your question, Thomas, that the C subunit itself can migrate away from the source of the initial signal. So that's part of the start, and it actually can, from imaging work, it can go reasonable distances away. It can even, under the right circumstances, go to the nucleus and then come back to the nucleus on its own. That's how it's one way of perpetuating the signal. The ERK kinase is part of a cascade, the so-called MAP kinase cascade, many of which you're familiar with, that's engaged typically by receptor tyrosine kinase that activate RAS, activate kinase RAF, another kinase called MEK, and then finally activate the ERK kinase. And so this is a canonical type of pathway that's found in many cell types that is also important in neurons. And I'll talk a little bit more about how this is regulated in a few minutes in terms of psychostimulants. But the way that these kinases engage each other, the RAF, the MEK, the ERK, is through the regulation of phosphorylation. So each of these kinases phosphorylates the second kinase, the downstream kinase, in a region near the active site that changes the structure of the active site that allows it to become active. So this is inactive ERK and this is active ERK. And it's phosphorylated at two residues. This threonine residue and this tyrosine residue both have to be phosphorylated for ERK to be activated. And when ERK is activated, it has many salicylic substrates, including the P90R6 kinase, other substrates, structural proteins like integrants. But for the sake of this presentation, the key substrates for ERK that we're going to talk about are transcription factors. So ERK itself can also migrate, once it's active, to the nucleus where it can phosphorylate transcription factors directly like ELF1 and other kinases that then phosphorylate PrEP. So we're going to talk about that. In contrast to the protein kinases, there are, actually let me go back. So it turns out this human kinome map comes about because one can take the sequences, the primary amino acid sequences of the kinases, and find in fact that a kinase is related by sequence and structure to other kinases. So all of these 500 or so kinases have the same overall orientation. They have a structure like this. It's a bilobate structure with beta strands in the N-terminus and alpha helices in the C-terminus. The active site in the middle, and the ATP binds here, and the substrate binds in this position, so it stretches along the junction between these two parts of the two lobes of the kinase. So if you look at any of these kinases, there may be little differences between them, but they all have this general conformation. In contrast, the pre-phosphatases have several different types of structures and several different types of mechanisms. So they clearly evolved differently from kinases to act as protein phosphatases. There are there's a subclass of serine 3 protein phosphatases. There's a distinct, mechanistically and structurally distinct group of tyrosine protein phosphatases. There are also other enzymes which I won't get into called atypical phosphatases, some interesting subclasses that we studied recently. Within the serine 3 phosphatases, there are two other subclasses, the type 1, which we already have a little bit about, the type 2 and the type 2B. 2A and 2B. 2B is also known as calcineurin. Some of you will have heard of that. Are structurally related to one another, and they are distinct from a fourth type called the 2C. And there's another subclass called the PPM subclass. This is called the PPP subclass. And there are related enzymes PP4, 5, 6, and 7 that belong to this PPP subclass. Within the tyrosine phosphatases, there are so-called receptor and non-receptor tyrosine phosphatases and dual-function phosphatases. And I'm going to talk a little bit about one particular tyrosine phosphatase. So it's sort of in this receptor subclass of these tyrosine phosphatases. And I won't talk about these guys. I've already mentioned that the key differences in these different subgroups is that phosphatases are mechanistically distinct from each other in contrast to kinases, which are one subclass of enzyme structure. I mentioned this a little bit, but these are the numbers again. About 32 of these PPP subclass and PPM subclass versus about 450 kinases. Whereas in the case of the tyrosine phosphatases, there are about 100 tyrosine phosphatases and about 100 tyrosine kinases, so they work more in balance with each other. Okay, so I'm going to elaborate a little bit more about the way that these phosphatases work, because I think it's important to make the distinction in terms of how they find their substrates within cells. So it turns out that the PP1, a catalytic subunit, actually has very diverse roles in cells because of its ability to interact with these so-called R subunits, which are variable, exchangeable, and bind one at a time. So one will find within cells PP1 engaged structurally with a number of these R subunits. So the guesses are that there are probably more than 200 or more of these R subunits just for PP1 itself. So immediately we actually think about PP1. When we think about PP1, we're not talking about PP1 as an individual entity, a catalytic subunit. We're talking about its dimeric state in concert with other PP1 binding proteins. And so there are probably within striatal neurons, there are probably several hundred PP1 binding proteins. So it's the regulatory subunits that are dictating the specificity, not the basic activity of the catalytic subunit. So that's a critical aspect to think about. The PP2A catalytic subunit also has diverse interactions with subunits. In this case, there are less of these regulatory subunits. In this case, they're called the B subunits, but they still have the ability to influence substrate specificity and localization within cells. The PP2A also has this additional invariant A subunit. It's just a structural subunit. Calcineurin protein is regulated by calcium and calmodulin through the ability of calcium calmodulin to bind to the catalytic subunit of the calcineurin, and this enzyme is maintained in an inactive form. It's activated enormously by the interaction with calcium. So when we're talking about calcium-dependent phosphorylation, calcium-dependent dephosphorylation, we're usually talking about the ability of this calcineurin to be activated by this calmodulin factor. And I'm not going to say much about PPC, but the key thing is that it's these regulatory subunits, particularly for PP1, that interact with PP1 and can either act as inhibitors directly or indirectly. They can dictate the enzyme localization and the substrate specificity. And I'll elaborate on that in the next couple of slides. So, so when we talk about protein phosphorylation, we usually think that the kinase is the thing that normally mediates activity. The kinase is the target for the second measure, and that's the thing that dictates the state of phosphorylation. But it turns out, in fact, particularly in terms of what we're going to talk about in terms of drugs of addiction and the effects that we have on striatal neurons, the mechanisms that regulate the phosphatases turn out to be almost as important, if not more important. So, in terms of protein phosphatases and similar transduction mechanisms, it's important not to think simply of phosphatases as a simple off switch. In fact, in many cases, phosphatases are an on switch, which may be sort of paradoxical to think that way. Particularly, good examples are the Sark family kinases, which are important for many aspects of cell proliferation, but are also functioning in the CNS in a number of situations. But they actually have to be activated by dephosphorylation. So the first step in activation of the kinase is actually the action of the phosphatase. Similarly, kinases in the cell cycle are regulated the same way. The phosphatase and how the phosphatase sets a baseline state is really critical. And so you can imagine different scenarios. If you have a very high phosphatase activity, that's going to be overcoming that phosphatase activity is going to be the first thing you want to do to allow the kinase to have its action. And that's going to be the situation for the DARP32 protein that I'm going to talk about in a few minutes. So it may be critical to modulate the activity of the phosphatases to get the kinase activity to be functioning. When we think of the way that these phosphatase and kinase cascades work in cells, we often think of their ability to amplify signals. So again, getting back to the question about localization within the cell, one of the critical things is to take the activity from a local lagging gated G protein and take that cell and amplify the signal. And so one can then spread that signal to different parts of the cell. And so these kinase cascades and the interaction of kinases and phosphatases through those cascades is critical for both amplification of the signal, but also for creating, and I won't get into great detail here, but also creating switch-like states. So you can turn signals on and off by the right organization of these signal transduction cascades, and I can go give more detail on this, but basically the MAP kinase cascade is often formed of a switching mechanism. And so one thinks in terms of the mechanisms that modulate synaptic plasticity when you go with LTP or LTD from one state to another state. You may be asking kinases and phosphatases in these signaling pathways to actually change from an on state to an off state or vice versa. So the interrelationship with kinases and phosphatases is particularly important for setting the alpha sensitivity of these pathways. If you get really clever, you can start to use these kinases and phosphatases over time and design oscillators and clocks. So those are all very interesting. And again, I could get into that if you're interested. Yeah. Okay, so basically, I don't have a diagram to show. It's a little hard to explain, but basically, the circadian clock is set by the generation and degradation of transcription factors that are produced and then moved from the nucleus to the cytosol, and then phosphorylation mechanisms are then used to induce phosphorylation in a temporal fashion that then cause phosphorylation of the transcription factor, which then over time is either degraded or altered, goes back into the nucleus and then modulates the transcription of other genes, and this is all done in circadian clock about 25 hours or so. And the key aspects of that are production of the transcription and the degradation of the transcription factors, but the engagement of putting kinases and phosphatases that modulate that process at different times in the circuit. And if you look in the textbook, you'll find that, or I can explain it. And I already elaborated on this point. Okay, so one of the key things in terms of kinases and phosphatases is how do they work? And I realize I'm probably running a little bit behind time here, so I'm going to go through this reasonably quickly. How do they recognize the targets and their target substrates in the cells? And so I already alluded to this, but kinases work by recognizing these phosphorylation site sequences within within substrates. They're a target at the active site. So a lot of how kinases find their targets is dependent on this action. There are also what are called docking interactions, which are non-catalytic. Domains can interact and dock substrates through parts of the kinases that engage their substrate in the right place. There are also other modular adapters and proteins that target kinases in particular places in cells and subcellularly target them to those sites. And some of these sequences are, for example, protein kinase A recognizes these canonical basic residues in the targets, so-called basophilic, and there are other types of targets that one can find for other kinases. In contrast, I try to make this reasonably brief. The phosphatases work more by localization of their targeting subunits. And so when we talk about where phosphatases are within these medium-spiny neurons, it turns out they're highly enriched in the synaptic, near the synapse itself. So this is a cut through of one of these dendritic spines. Here's the dendrite, here's the neck of the spine, and this postsynaptic density here, presynaptic element of mitochondria here. So in fact, this black stain represents in the medium spiny neuron is the localization of protein phosphatase 1. So the localization of the phosphatase is very, very critical. And it's highly enriched right at the site where the dopamine and the glutamate receptor signaling would be initiated. So you can imagine by positioning a phosphatase here that you'd have a lot of control over how dopamine and glutamate would function at this part of the cell. So again, going back to the question of localization, a lot of the initial regulation is occurring right in these synapses, but then it's perpetuated to the rest of the cell. Okay, I'm going to skip through this part because we don't have time. Okay. Okay, so hopefully I went over some of that a little bit quickly, but I want to shift now really to the main part of the talk, which is main part of the talk relevant to drugs of abuse, which is how these particular striatal proteins and the examples I've given of kinases, phosphatase, etc., should become obvious when I start to talk about the function of this DARP32 protein and how we think it's mediating the effects of cocaine. So the original description of DARP32 came about many years ago. It's now we discovered DARP more than 30 years ago from what we call proteomic screen, where we were looking for enrichment of cyclic AMP regulated phosphoproteins. So I alluded to the fact that the original description of cyclic AMP being generated by dopamine receptors came from just simple biochemistry by homogenizing different parts of the brain. And we took this very similar approach many years ago to look for what might be the targets for cyclic AMP-dependent protein kinase that might be highly enriched in the striatum. And we identified 3 proteins at that time, a protein called DARP32, another protein we called ARP21, another protein we called ARP16. Now I'm going to talk only about the DARP32 protein, we published on these other proteins, but you can see from the immunostain here that DARP32 is highly localized relative to the rest of the brain in these medium spiny neurons. I'm not going to show the details of that of this striatum. It's present in all medium spiny neurons and very high concentrations, probably anywhere near 100 micromolar or so. The other proteins, ARP21 protein and ARP16 proteins, are shown in the white stain in these 2 sections, are both also highly enriched in these same neurons. And we've worked on those and we've purified them, we've characterized how they work, and the amazing thing is that, in fact, each of these proteins, DARP32, the ARP21 protein we now call RCS, the ARP16 protein, all work by virtue of their ability to modulate protein phosphatases, in particular serine 3 phosphatases. So when we engage the D1 receptor, as shown here, we generate cyclic AMP, we activate protein kinase A. This phosphorylates DARP at a particular site that then engages PP1 and inhibits it dramatically. But in parallel, these other 2 proteins, the RCS protein and ARP16, are also phosphorylated by PK, and these then regulate either indirectly the calcineurin phosphatase or directly the protein phosphatase 2A. So for some reason, in these striatal neurons, they've evolved to highly express these 3 proteins that control essentially all of the serine, or virtually all of the serine 3 phosphatase activity in these neurons. And the reasons evolutionary for why this is not really clear at the moment, but I'll try to speculate a little bit at the end. So we already had a little bit about DARP, but how does DARP work? So DARP is really a signal amplifier. So the idea is that we have dopamine receptors, we activate cyclic AMP, activate protein kinase A. Protein kinase A phosphorylates a bunch of substrates in the cell, many hundreds of substrates. But in striatal neurons in particular, where DARP's highly expressed, DARP becomes phosphorylated on a particular site, 3D34. And this turns it into a potent inhibitor of protein phosphatase 1. Protein phosphatase 1 then is this phosphatase that dephosphorylates many of these same substrates that PKA phosphorylated. So by activating the kinase and inhibiting the phosphatase at the same time, then you get a large, huge amplification of phosphorylation. If you knock out DARP32 and stimulate here, you get much, much less phosphorylation. The signaling through the kinase still works, but the actual functional phosphorylation that's relevant to the cell is completely dependent on this phosphatase inhibition pathway. Okay? So here, the phosphatase is actually basically very active, suppresses the phosphorylation. If you activate only kinase, it's not enough to overcome that phosphatase activity. You have to engage the phosphatase inhibitor through the DARP32 mechanism. And together you get the right balance of activity. So we've studied this protein for many years, and I won't go into all the details. This is in review articles you can find, but we study many of the inputs, dopamine inputs, glutamate inputs, and many outputs. DARP has PP1 and has many substrates in the cell, both in the cell membrane, but also in the cell nucleus. I'm going to focus on that in the next few slides. Okay. Let's skip over this, I think, a little bit. So kind of back to where we are at the beginning. So basically what I'm going to now switch gears and talk about how the dark protein we think is working within these initial and very initial and slightly longer term events that mediate these initial effects of drugs of abuse on these pathways and lead to changes in gene expression over this period of hours to potentially longer periods of time. Okay. And so, right, so where are we going to start? So basically, we're going to start with ERK, and then we're going to switch and talk a little bit about PKA and how dopamine and cyclic-AMP and PKA can influence ERK signals. So if one, this has already been alluded to, but if one gives a psychostimulant such as amphetamine or cocaine acutely to a mouse or a rat, one of the first things one sees is that the phosphorylation of substrates within these medium spiny neurons in the striatum is increased. So basically, we look at phosphorylation of ERK, there's very little phosphorylation. But if you stimulate with amphetamine, in about 15 minutes, you get very nice increased phosphorylation of ERK that occurs in a subset of medium spiny neurons, the D1 type medium spiny neurons. We also know that if you inhibit the ERK activation through inhibitors of the MEK step of the ERK cascade, then we can block behavioral responses to psychostimulants as shown here in this sensitization experiment. So basically we get phosphorylation of ERK, and we need ERK for behavioral activity. The question is, how is ERK phosphorylation regulated? And so what we know is that glutamate itself, by engagement of NMDA receptors, can activate a calcium-dependent cascade that can then lead to activation of this ERK cascade, which I've mentioned before. ERK is then activated and then can move to the nucleus and phosphorylate proteins like ALK and other kinases that can then in turn phosphorylate the rest. But how might dopamine be influencing ERK signaling through the D1 pathway? So in these experiments, which were done in a paper from Beaujean when he was in the Neuronal lab a number of years ago, we were interested in the role of DARP32. And so if you look in the exact same sections for phosphorylation of DARP32, what you see is that basically there is DARP32 phosphorylation. But again, when you stimulate with amphetamine, you see increased phosphorylation of DARP32. And the merge shows that this is quite robust, but in fact, it turns out that the phosphorylation of ERK only occurs within a subset of the phospho-DARP positive cells. So there are many more D1 DARP cells than the cells that are phosphorylated in ERK. So ERK is going to, we think, is dependent perhaps downstream of the DARP pathway in a manner I'm alluding to a few slides ago. This is not specific just for amphetamine. You see the same thing for cocaine. Cocaine stimulates phosphorylation of ERK in these pathways. It's the same for cannabinoids, for opioids, and for nicotine. And it turns out that if we look at the knockout mice for DARP32, either a total null for DARP32 or a selective 3-34 alanine mutant mouse, we find that in fact the phosphorylation of ERK in response to these various drugs of abuse is either completely attenuated or partially attenuated in the DARP32 knockout mice. So we know that DARP is needed, and within the cells that DARP is phosphorylated, ERK is phosphorylated in a subset of these cells. Similarly, if we look at downstream targets for ERK, such as crab or elk, we also show that these are completely dependent. The response in this case to cocaine is for 10 minutes is also dependent on DARP32. In this case, we use a 3-34 alanine mutant mice rather than the null. And if we look at the behavioral consequences in the same sensitization protocol that was used to show the effect of the MEK inhibitor, we see the same ability of the 3-34 alanine mouse to suppress desensitization to cocaine shown in these animals. So we're pretty sure then that the DARP32 at this 3-34 site is able to mediate the effects of dopamine acting through the cyclic AMP PKA pathway. and that this targets somewhere in this pathway, but even though again the ERK pathway is engaged, you really need to inhibit protein phosphatase to get full activation of this pathway and mediating effects in the nucleus. So how exactly is this going on? So again, we can use the same sort of approach using combinations of knockout mice and biochemistry to look at that. And here what we're really looking at is mechanisms of signal amplification and signal selection, which I've alluded to earlier. So it turns out that ERK is phosphorylated by this upstream kinase MEK, but it's dephosphorylated by two types of phosphatases. As I mentioned, there's a threonine that's phosphorylated in the ERK active site, and there's a threonine that's phosphorylated in the ERK active site. And we were particularly interested in a tyrosine phosphatase called STEP, which is striatal enriched tyrosine phosphatase, which is co-localized within these medium spiny neurons as the ERK signal itself. So we thought perhaps the STEP tyrosine phosphatase might be important in regulating the dephosphorylation of ERK. And we already knew that this STEP comes in two flavors, the STEP 61 and 46. Both of them have this phosphatase domain, but they also contain this thing called a kinase interaction motif that's important for their ability to interact with ERK and recruit ERK so that the tyrosine phosphatase can dephosphorylate the tyrosine of ERK. Moreover, we know that the phosphorylation of this KIM domain can occur through pKa itself. We've done this work in collaboration with the Lombroso lab over a number of years before, so we thought perhaps these things are all working together in a coordinated fashion to modulate the ERK pathway, and that's how DARP is working through PP1. So indeed, we proposed this pathway then that not only DARP is involved in the regulation, but also pKa can phosphorylate the STEP protein, and that this would then suppress the activity of STEP, and this would also then lead to amplification of this tyrosine signal in ERK protein. So we did a number of biochemical studies to try to connect these things together. And this is an example of some of the Western blotting studies we did, and these are done looking at the phosphorylation of the STEP protein through this gel mobility shift, either of the STEP 61 or of the STEP 46, or by Western blotting with a phospho-STEP antibody. What we can see is if you stimulate, in this case with amphetamine, you see an increased phosphorylation over a very, again, 10-15 minute time frame. But if we look at the phosphorylation in the DARP32 knockout mice, we find that you don't see this regulation. So this is quantitative over here. Moreover, if we look at a different component of the pathway, the MEK protein, the upstream target for ERK, we also find that its phosphorylation is regulated by amphetamine, and it's also influenced by the DARP32 knockout mice, such that so DARP seems to be affecting not only the STEP protein but MEK itself. So if we take this pathway then and put all these different elements together, what we see is we go from these two pathways that we've already talked about, the ERK cascade and the D1 dopamine DARP cascade, and then we can start to put really define detail on the pathway. So in fact, there are multiple levels where the DARP pathway can engage through regulation of the upstream kinase for ERK, the MEK kinase, and also through the STEP protein, it can suppress the ability of PP1 to dephosphorylate STEP and engage STEP in the inactive form. And so this is, already we've taken a number of these components, and they're really, the critical aspect is the fact that they all have to work in concert. If we take out one of the components, the DARP pathway or the STEP component, we will not see the appropriate activation of ERK. So it's really a combination of the different elements of phosphatase inhibition in particular, which is critical for activating ERK kinase. So hopefully that sort of reinforces the point that the phosphatases are really the key area where we need to engage those phosphatases. We need to inhibit these phosphatases within striatal neurons to get the proper and appropriate regulation of the ERK cascade that can then go on and modulate nuclear events. So we've gone about an hour or so, and I think maybe I'll talk maybe 5-10 more minutes, and then we can elaborate on any questions after that. Okay, so what does ERK do when it goes to the nucleus? I've already shown this in small aspects of the cartoons, but ERK can regulate transcription factors. It can regulate other kinases. This is a kinase called MSK1 that can phosphorylate things like histones, to have epigenetic effects, phosphorylate Krebs, to have transcriptional effects. And work from the Yale lab and others have shown by more selectively looking at targets downstream of these different events, that these transcriptional changes can directly lead to specific aspects of psychostimulants that are mediated by distinct downstream pathways. And this has all been published, and if you're interested in looking in more detail of this, you should go and look at those papers, and I can give you the references that's needed. But this is this is just okay. I'm getting really out of order here. Let me back up. So we're obviously interested in dopamine signaling through D1 receptors, glutamate signaling, and clearly the engagement of this pathway only occurs if one engages both the dopamine signaling and the glutamate signaling at the same time. And so this has led the Yale lab in particular. This is from a review article that Jean-Marc Giraud wrote a few years ago, that in fact this dopamine signaling through and the regulation of ERK can be described as a potential so-called logical AND gate for signaling, such that you only engage this pathway if you have the right signal through the glutamate pathway, which is responding potentially to external and internal potential information, and to dopamine signals, which are responding to these reward prediction error signals. And only if you engage both glutamate and dopamine at the same time, you get activation. If you have one without the other, either the glutamate signal or the dopamine signal, you're not going to get this appropriate activation of ERK within these striatal neurons, and you really need those two pathways to occur concurrently to get the full activity of ERK, and therefore the full permissive state to allow the synaptic plasticity events to occur. And so, in terms of a circuit level type of analysis, one can think of dopamine influencing aspects of signaling in these striatal neurons at a level which has the ability to modulate selectively inputs that are glutamatergic that are coming from cortex and be relayed through striatum. And so when dopamine neurons are activated, they can co-activate at their concurrent time and place interactions with glutamate, but this may only occur selectively at specific medium spiny neurons that are appropriately engaged by the concurrent activation of the dopamine and the glutamate signal. And this can then strengthen selectively signaling through a particular subset of neurons within this temporal loop that flows back to cortex, and for which the reward and motor-based pathways in the striatum are particularly important for. But the key aspect is the concurrent regulation of the ERK cascade within these individual neurons in the striatum. So I'm going to move on and just talk a little bit more about DARP32 signaling. And again, this relates to the question that was brought up earlier, but really, where is the signal generated and where does it go to and how long does it last, the signal duration of the timing element. And for this, I'm going to turn to a different aspect of DARP32, which is a pathway that we discovered again in collaboration with the Yale lab, where nuclear functioning of DARP32 is critical. So this is a similar slide to the ones I showed before, but when we were looking carefully at the DARP32 phosphorylation and localization within medium spiny neurons, in response to cocaine, we made, and it may not be very clear from this slide, but we made this sort of subtle observation is that most of the DART was in the cytosol and not in the nucleus in the saline treated animals. But when we treated with cocaine for 10 minutes, most of the DART actually became nuclear. It's a little hard to see here, but there was a significant migration. We used biochemical methods and other imaging methods to show, in fact, there was localization of DART32 to the nucleus. So this talks a little bit to the question which was raised. So the initial signals are occurring at the dopamine receptors themselves. Within 10 minutes, the signal can actually move to the cytosol. We think, and I'll show you the details of what we think it occurs, causes changes of phosphorylation of DART that then causes the DART32 to, on mass, be much more abundant in the nucleus. So these are quite rapid and sustained events that can mediate significant changes in the localization of signal transduction through these elaborate neurons. And this occurs rapidly and is maintained over a period of an hour and a half or so, and then it gradually falls back to baseline. Show this. So basically, this occurs with other drugs and other types of treatments. So it's a pretty responsive system to psychostimulants and other drugs. So we're interested to know how this occurs, and this is a slightly more complex view of DART32, which is phosphorylated by PKA at the screening 34 site. It's phosphorylation of this site, which mediates the inhibition of PP1, as I showed earlier. But it turns out there are other sites of phosphorylation of DART, which I won't get into all the details of, but one particular site is the serine 97 site that's basically phosphorylated by a kinase called kinase 2. And so it turned out that this phosphorylation event is particularly important for regulating the nuclear translocation of DART32. And we went about doing this in different ways through biochemical experiments or in cell culture with GFP tagged DART, which this slide is taken. And we asked the question, which of the phosphorylation sites in the DART32 molecule were important for regulation of this nuclear translocation? So we were pretty sure that, and we set this up in culture so we can stimulate with a D1 agonist. We get nice translocation of DART32 to the nucleus very rapidly within minutes of treatment with a D1 agonist. And we were pretty sure it was going to be dependent on this 3e34 site. But if we mutate the 3e34 to alanine, we still get a nice nuclear translocation. We mutate some of these other sites, this 375 site or this serine 130 site, there was still nice nuclear translocation. But if we mutate this serine 97 site, an alanine residue, so it can no longer be phosphorylated, what we saw was a basal increase in the nuclear localization of DART that was no longer modulated by the D1 agonist short. So clearly this site somehow is influencing the DART translocation. And it turns out it's a fairly complicated story, and I'll just try to reduce it to a couple of cartoons. But what we think is that normally DART32 has the ability to be to move between the cytosol and the nucleus. Basically, the serine 97 is phosphorylated by active kinase CK2, and it's in the cytosol. But when it's in the dephosphorylated state, it can translocate and be retained within the nucleus. And in fact, what we now know is that there's a continuous shuttle of this protein in and out of the nucleus. And it turns out many proteins, in fact, have the ability to go to the nucleus, but they're frequently removed from the nucleus by a sequence which is called a nuclear export signal. So in the case of DART, it has a very, very active nuclear export signal, which is adjacent to the serine 97 site. So we think in the presence of phosphorylation of the serine 97 and the adjacent nuclear export signal, the steady state is actually for DART to be localized almost exclusively in the cytosol. But if we modulate the phosphorylation of this, particularly by dephosphorylation in a PKA-dependent fashion, what we get is dephosphorylation of that site. The protein is then moves to involved to the nucleus, and it cannot be removed from the nucleus because it's not phosphorylated on the site. Now we went on to look in some detail at the mechanism, and it's a lot of detail here, but it involves the ability of cyclic AMP kinase to regulate this PP2A phosphatase. And it turns out we had found that one of the subunits, these B subunits, was phosphorylated. And so the mechanism then is that PKA with slightly slower kinetics than the initial activation of phosphorylation of DARP32 itself occurs as a secondary event. So initially DARP is phosphorylated by PKA, inhibits PP1 in the cytosol, but then slower, a little bit slower, PKA can phosphorylate this phosphatase on the B subunit. The phosphatase is then engaged, selectively dephosphorylates the serine 97 residue, and then the DARP32 can move to the nucleus. And so let's skip over this and just go back to the cartoon. So here's basically the same thing again. So initially, DARP is phosphorylated in response to cyclic AMP D1 acting in D1 containing neurons. It phosphorylates the 334 to cause an active form of DARP32 that can then go and engage PP1 in the cytosol. And probably initially the effects are very local to the synapse. They're probably occurring on the PP1 that's right at the synapse. That phosphatase is initially inhibited. The DARP can then, in this active form, migrate into the cytosol, and that's what we measure by immunocytochemistry or biochemistry. It's the cytosolic DARP initially that you see in the cytosol. But then, through the slightly slower event, the DARP can be dephosphorylated at the serine 97 site by this other phosphatase. And it's then, the DARP then is still phosphorylated on the 334. This can shifts in the nucleus, and therefore we now get inhibition of nuclear phosphat PP1 as opposed to cytosolic PP1. When we inhibit PP1 in the nucleus, we have a different set of events. We affect things like histones. We affect things like kinases that phosphorylate these histones. We affect the ability of PP1 to dephosphorylate CREB. So CREB and other transcription factors are selectively dephosphorylated by PP1. And then we can then get changes in gene expression, gene epigenetics, and long-term transcriptional events that are maintained in response to these initial signals, which engage very locally the D1 receptor and local changes like cyclic AMP. And so all of this, I think, is in concert is really a timing mechanism that selects strong signals in response in these neurons, as opposed to weak signals that may only engage some of these more initial aspects of the signaling. It's only when you get these things occurring concurrently in response to dopamine and glutamate in the right time in the right place that you can engage these longer term signals, because these phosphorylation events need ERK, which is not present in this cartoon for 6 seconds. So I think I'm going to stop at this point, and then you guys can ask questions. I have a couple of comments that a couple of more things I maybe elaborate on if you don't have questions, but I'll stop there and let you ask questions. So I don't know if I answered your question, but I tried to answer it. Maybe you can ask it more. But help if somebody else stuck my head up.
Nice overview. I was wondering if you know what the DARP does in the axon, because there is DARP in the axons as well, right? So
yeah.
Do you know it has a totally different role, or?
That's a good question. I mean, we've never really looked at that in a systematic way. DARP, if you look at this, if you look at the immunocytochemistry of DARP, you see terminals. There was a little bit of work done looking at neurotransmitter release, and as I remember, using synaptosome preps and looking at DARP knockout mice, and there was some evidence that there were changes in neurotransmitter release, if I remember correctly. There's also data from the DARP32 knockout mice, it's old data, where there was, if one looked at amphetamine toxicity types of paradigms, that there seemed to be circuit effects of DARP on the effects of methamphetamine to cause degeneration of some of the dopamine neurons, and so the thought was that DARP somehow was influencing local events at those terminals in the nigra. Those would presumably be the D1 cells, the direct pathway neurons. But to actually selectively ablate DARP in the axon is not an easy thing to do. When we look at or any protein for that matter, unless there's a specific targeting event, it's hard to perturb that. We have no evidence of anything within the DARP sequence that would target selectively to axons or not. What we've looked to kind of turn your question a little bit more around. We did look more in the dendrites and the dendritic spines, and so the data was never published. It was a little hard to quantitate it exactly, but I showed you one view of where we think most of the PP1 is in the dendritic spine. There's a lot of PP1 in the dendritic spine. We know why the PP1 is in the dendritic spine, because it's targeted by another protein, or actually two proteins. They're called spinophyllin and rabin, and they're highly enriched in dendritic spines, and they recruit the PP1 to the spine, both in the in striatal neurons as well as in other neurons in the frontal cortex and hippocampus. And if we look carefully, there seems to be an inverse gradient between the DARP expression and the spinophyllin expression within the spine themselves. So that sort of makes sense in as much as the PP1 can only bind one of these subunits at a time. So the region that PP1 binds to DARP engages a small part of the N-terminus. I didn't get into the details. There's a canonical motif in the N-terminus of DARP that's the same canonical motif that's found in many of these hundreds of PP1 binding proteins. So only one can engage at a time because there's not enough room. And so the PP1 that's bound to spinophyllin cannot bind to DARP. So when we look, we saw an inverse gradient relationship between the spinophyllin stain and the DARP stain. And we published a little bit on this, but basically what we think is that so what we think is that one of the first things that, and this was kind of what I was alluding to, I'll see if there is a slide back. I was alluding to earlier, but yeah, this slide. So here's PP1. PP1 is bound to the protein spinophyllin, and we think it's engaged and it's localized to the spine because the spinophyllin protein can bind to actin, which is a predominant cytoskeletal protein in the spine. And so the PP1 is maintained in collaboration with spinophyllin in the active form. So the PP1 it binds this, but it's active. Once we get engagement of the dopamine receptor, we can activate protein kinase A, and we know that actually phosphorylates spinophyllin protein and loosens it up from the actin. We published this a number of years ago in this paper. And in this paper, we elaborated on the fact that we have other data that we put in this in another paper where we think this form of the protein is then sensitive to phospho-DARP. But DARP works by both engaging the active site but also knocking off the spinophyllin. And this is all happening locally in the synapse. We did some electrophysiology and other things that showed that we perturb this interaction and synapses that had very rapid effects in electrophysiology. So we know more about what happens in the dendrite, but in terms of the axon, it's an open question. Next.
Does SNAP2 act as a signal amplifier in other brain regions as well, like for example in the cerebellum?
Yeah, that's so DARP is present in other brain regions. It's not present in cerebellum. It's present in some hippocampal neurons. I mean, maybe a little bit, but not much that I know. I mean, unless you have evidence.
It's just sensory isolated.
Okay. There are clearly neurons in frontal cortex that have DARP. There's DARP in hippocampus, in pyramidal neurons. And a little bit of work was done in hippocampus, looking at some LTP and LTD type phenomena, and there was no effect of knocking it out. And also there was work done with a homologue of DARP called inhibitor 1, which is also in the hippocampus, and the combined knockout of DARP and I1 didn't seem to have much of a effect. So if you look in the cells in the in other parts of the brain where you see DARP, what you tend to see is, as opposed to the striatum where all medium spiny neurons contain almost equal amounts of DARP as far as we can determine, you see selectively highly enriched cells that light up with DARP, and then other cells don't. So the answer is kind of a maybe answer. If you think of the distribution of total DARP, you would look and say, well, there's not much there, so therefore it's not going to be working as a signal amplifier. But it seems to be in specific cells, so there in those specific cells that it could have some capacity. The problem is it's really hard to study those cells. We don't have markers for them. The person who did that work, Charlie Wilmette, when he tried a bunch of different markers, it was not clear exactly what they were, what they might be co-staining with. Retrospective, or not retrospectively, but in terms of moving forward, it might be useful to take some drivers, mice drivers, and see if we can knock out some of those DARP stain, so to see if we can get some more information. But I think there is some evidence that DARP can function in other parts of the brain at a biochemical level, but the signals that you get from the outputs are just not as robust. If you look at it from theoretical considerations, in striatal neurons, the amount of DARP is somewhere in the region of 50 to 100 micromolar. The amount of the phosphatase targets is about 15 micromolar. So there's a huge excess of DARP over the phosphatase in the medium spiny neurons. And so even if you and biochemically, if you stimulate phosphorylation of DARP at 3 and 34 with a strong signal like cocaine, or in a slice with forskolin or something like that, as was mentioned, you get about 10 to 20% of the DARP is phosphorylated. So 10 to 20% of 100 micromolar is enough to suppress the activity of virtually all of this PP1 in the cell. So from a theoretical perspective, we did look at the distribution of phosphatases and their activity and levels of the different isoforms for PP1. And they seem to be pretty uniformly distributed through the brain as through all tissues in the body. So the same amount of phosphatases there. But there's probably going to be less DARP, except in a few cells where it's more expressed. And so the dynamics and the kinetics of the phosphorylation, dephosphorylation events are going to be influenced by those two factors. So it's a kind of a, you probably say that if you did it from a theoretical perspective, there's less likelihood that DARP would have ability to modulate in those cells, but we'd have to be careful to look at specific cells. And I suspect if you would over record, maybe we should make a DARP GFP mouse, which we never did, and we was thought about doing it, but then go in and then selectively look at those cells and record from them and see if there's particular properties, and then go from there. But it's obviously involved process.
Is the mechanism the same in other dopamine sensitive organs, for example, cardiovascular system?
That's a good question. So it turns out that the question was, let me paraphrase. The question was, does this dopamine sensitive signaling pathway work in other tissues and organs? And the answer is yes. So DARP is expressed in the kidney, and DARP and D1-like and D2 receptors are expressed in the kidney. So part of the salt, elements in salt, is actually mediated through the DARP pathway. So work has been done by Anita Perrier in Sweden a number of years ago, showing that there are phenotypes in DARP if you knock out DARP on natriuresis. And there's some details of where the DARP is. I can't remember the details now. It's been a long time since I looked at the papers, but there's specific expression of DARP in certain cells in the kidney that have the dopamine responsiveness. It's a little bit more complicated in terms of D1, D2. The receptor, if I remember correctly, exactly how the receptors respond, whether they're exactly the same as the neuronal ones is still not clear. There's some really odd things. So in certain types of fat cells from certain species, some of them express DARP, and other do not express DARP. They express this inhibitor 1 homologue. But the idea is that if DARP is expressed, there's a capacity to be engaged by cyclic AMP signaling. And there's a little bit of work was done in I think the problem was the mouse didn't normally express DARP in those cells. It was in like rabbit and some other species. So the answer is yes, there is DARP. There's also, from a more conceptual perspective, there's also the basic signaling elements are found in other cell types. And so if you look in muscle, there's very high levels of this inhibitor 1 molecule I mentioned, which is a different gene product, but in the first 40 amino acids, it's almost identical to the first 40 amino acids of DARP. And so the original work on DARP was actually predated by work on this inhibitor 1 molecule, which is responsive to beta-adrenergic signals. And skeletal and cardiac muscle can be modulated through those pathways. And so if you knock out the inhibitor 1 gene, you see effects in signaling in those muscles. Okay.
This is a very general question. You focus part of your talk in the D1 receptor signaling, but I am very curious if there is any information about the role of the dopamine heterodimers in drug abuse, exposure, or addiction.
So I missed the last
in the dopamine heterodimers with drug abuse or addiction.
Dopamine heterodimers?
Yeah, heterodimers.
You mean D1 and D2 receptors?
Or
other receptors.
Yeah.
So there are a couple of things, and I showed a summary from a review from Susan George that came out last year, I think, in Quantitative Neuroscience or something like that. There are a number of people who are investigating those interactions. And you can sort of do it a couple of different ways. You can set up artificial cell systems which co-express those receptors and look at signaling events. But those, I think, are artificial. And so, if you're really going to study in real tissue in response to physiological stimuli, I think I don't think we really have the tools to discriminate necessarily, because in a typical cell it could contain, let's say, a D1 direct pathway neuron that contains mostly D1 receptors. About 5% or in that range seem to co-express D2 receptors. And the distribution of those seems to be quite random across different parts of the striatal. And so you really are looking for a probe that would be a distinct readout of something that was engaged only by D1, only by D2, or by the two together. And so technically, it's really hard to rule out where the signal is coming from. So there are people who are doing that, and they claim that there is selective engagement to this engagement of these dimers. But from my perspective, I'm not so clear that we have the right readouts. The other way to do the experiment is to use selective antagonists or selective agonists that would only engage the dimer versus not. And some of those are being developed, and there is data out on those. Again, I think it's maybe premature to say for sure what the details are. So I thought maybe you're going to ask a slightly different question, which is, I talked about mostly D1 cells, but DARP is in the D2 cells too, the indirect pathway neurons. And so what's going on there? And that's a whole other topic. But what we think there is that in fact there like others can probably comment on this, and maybe others will talk about this, but it also depends on where you are in the striatum. And the dorsal striatum is not the same as ventral striatum. The distribution of the D1 and D2 receptors is different. The co-localization is different. There's much more overlap between D1 and D2 receptor expression in the ventral striatum. Numbers go from about 5% to about 20%. There may be other effects through development that have different co-expression. What we do know is that in the D2 cells in dorsal striatum, where you can do these experiments a little bit more rigorously, that DARP is present, that D2 receptors cause dephosphorylation of DARP32 at 3 and 34 through the inhibition of adenyl cyclase by the mechanism which I mentioned. But then you ask the question, well, what's driving the generation of cyclic MP that maintains the levels of cyclic MP that would allow D2 receptors to work against? In that case, the cyclic MP is being generated tonically by adenosine A2A receptors, most likely. So what you have is a situation, at least in cells that you can label as either pure D1 or pure D2, completely opposing signal transduction pathways. One is cyclic MP increase and one is cyclic MP decrease, just for simplicity. The DARP32 system is working exactly the same way in each of them. So, and I can show you some data on this that if you're interested in. So again, the question is, how do you go about studying that? And so we heard from the presentation this morning that you can use these D1 and D2 GFP mice, which exist, and you can buy them or get them from your friends. And you can use, if you have a particularly good probe like phospho-S6 antibody, which is quite clean, or a phospho-ERK antibody, you can co-stain for GFP and you can see the signals in those cells. But unfortunately, there are not that many antibodies that are clean enough to do that. There are certainly some. Phospho-S6 is good, phospho-ERK is good, our phospho-DARP antibody is okay. There's some phospho-CREB antibodies out there and a few others, phosphohistone antibodies, which are good. So if you have the right tools and they work, you can interrogate those cells selectively. And if you look at the data that's there, what we know is that side by side, the D1 and the D2 cells are intermingled, and DARP32 phosphorylation at 3 and 34 will go up in the D1 cells and go down in the D2 cells, side by side. If you homogenize the tissue and run a Western blot, you get an average of the two signals that usually equates to virtually no change. Okay. And if you push the system hard enough with a lot of amphetamine and a lot of cocaine, or if you're in a tissue slice where you can push it with forskolin or some other pharmacologically, set up your assay so you favor the on signal as opposed to the off signal, you can get an increase in the signal. But normally in a mouse that's responding to more physiological signals, in fact, biochemically, the data would be averaged out. Okay. So that's a big challenge to get at studying that, and there are a few tools. But if you don't have those tools, you can't assume that what you're looking at is in one or the other. That's the bottom line. So, let's see if I can flip through these. Right. So this is kind of one example of the D1 cells and how we think they work. But in fact, in the D2 cells, I think all of the signal transduction machinery that involves DARP, phosphatases, ERK cascade, things that I talked about, and many other things which I didn't talk about, they actually coexist in both the direct pathway neurons and the indirect pathway neurons. And this is kind of a bit of a guess, really, but one of the reasons I think that the phosphatase regulation has evolved in the way it has in the D1 and the D2 containing neurons, and how this really, I mean, I only showed, as I alluded to, for phosphorylation, we are only looking at the iceberg thing, the tip of the iceberg normally, but I only showed you the simple version of the signaling pathways. Okay, if we actually take all the data we have on all the signaling pathways that we know something about, they get enormously complicated. with many, many factors, probably 50 components, and that's a simple model. Those 50 components are working in the direct pathway neurons and also in the indirect pathway neurons. And one of the reasons I think it's set up that way is, in fact, they have to do two different things. They have to respond one way in an on direction in the one set of cells, and they have to go in the other direction in the off cells. But they have to do it in a coordinated fashion. And then they have to reverse themselves in a coordinated fashion, so they have to be switched on and off in opposite ways. And so I think a lot of the signaling machinery has evolved particularly to allow key elements like glutamate, dopamine, but particularly dopamine, to work within the concert of the way that dopamine is released either tonically or physically at the dopamine terminals. And so I think there's a combination of just switching things on and off, and then switching them off not so strongly, and switching them on not so strongly, but switching them on very strongly when you need to engage more complex signaling to the nucleus. And so, probably not just me. I think most people in signal who study signal transduction would say the same thing. Signal transduction complexity is used for various things: integration of signals, amplification, designing of sensitive, ultra-sensitive switches, and the like. All of that probably happens too, but for some reason in the striatal neurons, they're doing a little bit more complicated signal processing that has to be coordinated into this larger network that involves the direct and indirect pathway neurons, and I think that's part of the reason for all this complexity.
You showed a slide about our translocating to the nucleus and interacting with ELK, MSK, and Krebs. Does that happen in a specific manner? Does it interact with specific downstream proteins in different cells, or is there anything known about that, or is it just a cascade?
So I'd say the best data that's localized those signals is, so you can look at phospho-ERK and you can look at phosphohistone H3 as a very clean antibody. I flashed past some slides that had it on it. So when you look carefully, and you can do this in mice if you're good at it, and you can do it in cultured neurons, so that you can manipulate expression of some of these factors. There seemed to be a pretty good correlation between phospho-DARK, phospho-ERK, and phospho-H3, in not just in a population level, but at cell-by-cell level. So I would say that probably that there's a basal level of activity that has to be engaged, and if you push the signaling pathways enough, you will engage many of the things which I talked about. I think, does that answer your question? So that I would say, yeah, I think the function of the signaling is to get to the endpoint, and so all the things that you would want to see would be the case. A slightly different take on that is, I wish we could do a little bit more, but we did a little bit of work with a mathematics group to try to model some of this stuff, and which is a whole other area of signaling, which has its own strengths and weaknesses. What came out of that, and in fact, several labs did that. The Yale lab did it, and a Japanese lab did it, and a group in Sweden did it. There were a bunch of modelings in the dark pathway that are actually quite interesting papers, because we all did the modeling differently. But the interesting thing was the result was the same. One was that dark was completely critical to all of the models. So that was good. But in our modeling, the way that we did the modeling was we didn't constrain the model by actually putting in any real values for rate constants or concentrations. What we did was to basically have inputs that were either 0 to 100% glutamate or 0 to 100% dopamine. And we basically allowed all of the parameters of all of the other components to be altered randomly, essentially. And so what we determined was that, in fact, the dark component of it, particularly one of the phosphorylation events, the serine 97 event, was critical for making the system robust. So the term that's used in mathematical modeling And what that sort of simply meant was that we could actually have some of the components of the pathway not conforming to what should be happening, but as long as we had the core elements right, the input and the output could be maintained. And so there is some flexibility in the system that not every component in a single cell type that engages these pathways would necessarily have to respond, but because you could push a little bit on another side of the pathway. But by and large, biochemically, what we see is a cell will be turned on or turned off.
You could clearly describe what happened after an acute cocaine treatment, but what happened after chronic treatment?
Okay, so obviously it depends on what the readout you want to look at. So in some cases, in this dark pathway, and there's these medium-spiny neurons signaling pathways that we talked about, and there was some data that was shown is that you can look at things, other behavioral outputs like locomotor sensitization, condition-based preference, other behavioral outputs which are sensitive to repeated delivery of psychostimulants, for example. Without getting into a lot of detail, some of which I don't remember off the top of my head, things start to get complicated. And so basically there's data out there, and I can give you the references, but if you take the DART32 molecule and mutate the 334 site, the 375 site, the serine 130 site individually in mice, and then do experiments in response to acute cocaine versus chronic, 14-day exposure or whatever, you see specific contributions of the phosphorylation sites. It's not easy to summarize because there's no easily consistent model you can come up with. But basically, there are effects on some of those more chronic parameters that you knock at 375, for example, that you don't see with 3D34. But I'd have to go back and look at all the data to tell you. So it's clear that these pathways are being engaged by chronic exposure to particular psychostimulants. In related experiments, and it's not related to DARP itself, we have looked at changes in the activity of other phosphorylation events that respond very clearly in distinct patterns. And so the parameters we sort of used were either an acute stimulation, a 14-day stimulation, followed by a drug removal time of usually 14 days, and then response to an acute stimulation, and try to compare all three cohorts. And what it looks like is that there seem to be longer-term changes probably in the phosphatase and kinases that are, particularly we're looking at some CDK5 targets that in fact, if you chronically treated every day for 14 days, and then you would see actually, and a critical thing here is when do you measure the phosphorylation event after the last day of injection, because you're going to have acute effects in response to those that would follow similar pathways. But if you wait for 24 hours or so there's no drug on board, you would often see no change. The phosphorylation of those targets would be the same as the baseline. But if you then challenge with an acute injection, you would see different kinetics to what you would see in the initial acute, naive acute animal. And so there's a number of examples in striatal signaling where you see things like that. And what we think are that a couple of things happen, and some of this is published, some of this is not, is that one can engage the expression of targets for, I know somebody's going to talk about some of the downstream epigenetic changes, but things like delta phos-B, which are induced by acute stimulation and can be increased in expression with chronic exposure, then affect the transcriptional profile of certain targets that are then maintained for long periods of time. So one of those is the CDK5 kinase, and so it can be upregulated, and that suppresses the action of DART. And so what we see is an increased CDK5 activity, an increased phosphorylation of DARP. So it's sort of simplistically, this cell is desensitizing the DARP pathway in the way that I described it. So you see phenomena like that, and obviously, and I didn't get into this at all, but in any G-protein coupled receptor signaling pathway, no matter where you start, from ligand engagement to endocytosis to all of the myriad mechanisms that are involved in receptor endocytosis recycling, basically all of the signaling is set up to desensitize. And so I think one of the interesting things about drugs and abuse and the effects that they have is that they tend to desensitize in a different way than they would to normal endogenous ligands that engage those receptors. So you see changes that are longer lasting and are maintained, and I can go through it for more specific examples. Questions? No? Good. More questions? Did I answer your question yet?
Still wondering, like, what is like the whole process of like the surface trafficking of the dopamine receptor. It's like the spatial organization of how close they are to sites of excellent transmission underlies a lot of the signal. So I'm just wondering what is known about the dynamics of the dopamine receptors on the surface. I mean, beyond endocytosis, just like what recruits them or what brings them into proximity of postsynaptic sites. Whether there are differences between different, let's say, whether the molecular composition of excitatory synapses makes a difference, whether they have dopamine receptors in proximity or not, so whether some synapses are more primed to contribute to this type of signaling. So I'm thinking about the dynamics and
so
continue.
Right, so part of that, I think, again, is really method dependent, right? So it's method and reagent dependent. So, I think it's still true that we don't really have good receptor antibodies for the probes for the dopamine receptor themselves, the D1 and D2. I mean, that was a big problem with a lot of the confusion in terms of expression for a long time, is we didn't have the good antibodies. And then there were some decent antibodies that worked for some people that were good enough to localize the dopamine receptors to these to spines and so on. But when you're talking about dynamics, you really have to be able to study that with the right, with the right imaging methods. And in that case, and often you have to go to reduced systems, simpler systems, culture systems. What I can say, that I remember off the top of my head, is that the Aperia lab has been using some fairly high resolution imaging methods to look at D1 receptor and NMDA receptor dynamics in adjacent to or in spines. And I don't remember all the data, but what it's simplistically is that clearly are interactions that occur, and there are ways to stabilize the expression, the co-expression of these two types of receptors in the right place in the spine. So there are people who are interested in this. Von Zastrow's lab is interested in some of the same questions. Clearly there are reserves of receptors that are not as functional, that can move from the plasma membrane close to the synaptic area of the synapse, and some of them can be retained there, obviously by accessory proteins and TARPs and things like that are critical. How the dopamine receptors or other GPCRs are co-engaged, I think, is still an area where there's a lot of work being done. I don't think we know specifically the mechanisms, but we know that, and unfortunately, from my perspective, we tend to do things at population level, so we can't do those experiments. And the people who do them with enough resolution can only look at individual synapses, and so there's a part in between is really, I think, the challenge. And, it's a challenge for how signals emanate. Particularly, you may have heard Dick Chen talk at Yale recently on how L-type calcium channel signals perpetuate the nucleus. So there's very clear L-type calcium channels at synapses can turn on Krebs very selectively. Somehow there's a mechanism that manages to convey calcium-dependent signals in a protected fashion from the synapse to the nucleus. And some of the same questions arise. How do you do that? It's really complicated because you would think, you know, in something like calcium, the signal would be dissipated by the endogenous mechanisms, the reuptake mechanisms, but yet it seems to be perpetuated. So those questions, I think, are really critical ones, and you know, we're kind of a little bit too far downstream to use things like DARP as a readout. Okay, thanks.